So,
All
Dr.
right, so yes, we're going to start off going over this lab exercise here in our fourth lab before we do the tests. We're going to be going over each answer. Any questions just from the beginning here?
Yeah, with osmosis, it uses the aquaporin, right, as a carrier,
Yes, it does, as a channel, yes.
as a channel rather, not a carrier.
Yeah,
It uses the aquaporin channel. So but that's still considered passive transport,
oh, it's definitely passive, yeah, because it's moving down its own gradient.
moving down its gradient, so it's passive.
Yes. Yes, it is. Okay, well, let's take that matching from the top. This is our exercise here, just a review of the last lab from lab 3. So we will be starting the new lab information, of course, after this, but I wanted to put this in there because it's a great review. I find it just strengthens what we should have known coming out of that third lab. So let's do the matching from this standpoint, reading the description and then going for the word. Describes a low solute environment, more water.
It's hypotonic.
There you go. Perfect. So with that description, it kind of tells you the answer, doesn't it? Low solute. Hypo means low, tonic means solute. There you go. There's your translation right there of that, what that means. And anytime we talk opposites, realize in a single compartment, if you've got low something, you're going to have high something else. So in this case, if we have low solute, it's going to be more water. Okay, that's always the case. That's just how it is. You don't even have to talk percentages. If it's low of one thing, it's going to be high of the other thing in that same compartment, in that same environment. So if we're talking inside the cell or outside the cell, each of those represent two different compartments. That'll be the case in every compartment. The next one, passive transport of water-soluble solutes using membrane proteins.
Facilitated fusion.
Very good. How about passive transport specifically of water molecules themselves down their gradient?
Osmosis.
Excellent, osmosis. So just to clear things up, osmosis can be known as a type of facilitated, to be honest, but we give it its own word for osmosis because it's just that special. And it has its own carrier, doesn't it? Or excuse me, its own channel. Yeah, there you go, the aquaporin. Perfect, perfect, perfect. So it is a membrane protein. It's called an aquaporin. It is considered facilitated if you get down to it because it's using a membrane protein. Anything that uses a membrane protein can be known as facilitated in a way, okay? Or at least it's known as carrier mediated is how they like to call it. I know that's a loose term used in the textbook, but when we use ATP versus no ATP, that's when we start talking about active and passive. So that is the difference. So don't be confused by that. Okay, let's move on. Next one, major extracellular cation based on active transport. So before we answer, what is active transport? What's the quality of that sodium potassium pump? How many sodiums are moving out versus potassiums, or what is going in and out?
3 sodium out, 2 potassium in.
Right. So the sodiums are going out, which makes sodium our major extracellular cation. So let that be the best way to remember that is based on that sodium potassium pump, because it doesn't matter what's going on, unless that cell is acting on an impulse, sodium ions will constantly be pumped out with that sodium potassium pump, creating a larger amount of sodium ions on the outside of the cell. Perfect. Describes a high solute, less water environment. How about that one?
Hypertonic.
Yeah. Again, how do we describe that? High solute. Hyper means high, tonic means solute. Specifically from the textbook, it means impermeable solutes. But guess what? In this question, we don't care, to be honest. I mean, let's just make it easy. Sometimes there are specifics like that, but in this question, high solute means hypertonic. It could mean high osmolality also. If you're confused by that, it's the same old thing. The only difference is if the solute is permeable or impermeable. Tonicity refers to impermeable solutes if you really want to get down to it. But in this question, does it mention permeability? Nah. It's just high solute, so it's hypertonic. And anything high in solute is going to want to attract water. It's like, come here, water. I got you in my eyesight. It's going to pull water towards it. So when you put a cell like down here, down below on the quiz here, you put a cell in a hypertonic solution where the solute is higher on the outside, that water is going to chase that solute right out of that cell, and that cell is going to shrink. So remember, water always moves, or you could say attracted to the higher solute. And in this case, it's the solution, it's the environment that the cell is being put in. I think that's what gets people mixed up. So
Yes.
pay attention to those words. It's always the solution that the cell is put in, not what's going on inside the cell. We just want to see how that cell is going to behave. It's like taking your little toddler, your little 3 or 4-year-old to the first birthday party and seeing if they're going to be social or shy. How are they going to react in that environment? That's what's happening. Okay, next up, passive transport of lipid soluble solutes.
Simple diffusion.
There you go. Lipid soluble means they're nonpolar. And guess what that cell membrane is? Largely nonpolar. Due to all those fatty acid tails, they create a nonpolar fence. Therefore, water-soluble molecules will have to use facilitated methods to get through. They need a protein, right, to get through. But lipid-soluble solutes, they slip right on through. So we would expect lipids, steroids, and nonpolar gases like O2, CO2, methane, those examples of things that are nonpolar that would pass through. So simple diffusion is for lipid-soluble, not water. Water tries to use simple, and it may succeed, very small amount of time accidentally, but water is going to use what to get across?
Osmosis.
It's going to use osmosis, which is moving down its gradient passively, and it's going to use that aquaporin. So yeah, get all that straight before the test, for sure. Or rewind this video and hear me say it for the 3,000th time. And I don't say that sarcastically. I laugh about it because I know how stubborn I was getting this material. And truthfully, like I've said before, the teacher can say it 5,000 times, and it's still going to go right over your head until you own up, right? Put that phone down, tell that boyfriend or girlfriend to hold on, I got work to do, right? Put yourself in another room away from your kids, whatever it takes, and write this stuff down on paper. Figure it out, right? Make it a personal goal to figure it out, and it's yours forever once you figure it out. Somebody can teach you all day long, but until you figure out something on your own, that's where the magic is. And guys, that is the only way I'm sitting here teaching you this stuff. I feel like I didn't learn it in school. I'll put it that way. Feels like I learned it on my own because I'm just that stubborn of a person, I guess. I don't understand the way other people talk a lot of times, especially stuffy professors that really they only care about their own life. I've had many of those. All right, last one, passive transport, or excuse me, major intracellular cation. So intracellular, dictated by that active transport. What do we get?
Potassium.
There you go. So that's our last one. So yeah, those are all your answers to those questions. Great, great, great. And if you're still weak on that, possibly a few of you got some things mixed up between water and stuff, no problem. Just fix it today. Reprint the quiz, take it again till you got it. That's how you learn. That's the best way to learn, is finding out what you get wrong. All right, so I'm gonna clear these answers so I can, well, actually I can leave that there for a moment. Let me just work on something else here. How about number 8, active transport is up or down the gradient? Up. So what that really means is that we're moving solutes from an area where there's low concentration, possibly inside the cell, it could be either or, to an area that's in higher concentration. So our good example is that sodium potassium pump. It's going to be moving sodium ions where they're low inside the cell to where they're high outside the cell. By the way, that's what makes sodium the most major extracellular cation right there, is going up the gradient. Passive is Down the gradient. Yep, that's where you could say that's its energy, but in a sense, there is no energy in the form of ATP. But that's the natural flow that happens when you fall downhill from an area of high concentration to low concentration. It just happens on its own. There's no energy push needed. It's just a natural occurrence to go from high to low. Just like it's easy to have $100 and spend it all, right? Go down from 100 to $1. Or you can blow 100 easy nowadays, right? Half of that sometimes just to take the family to Chick-fil-A. But it may be a little harder to get from that dollar to reach up back to 100. That takes some energy, doesn't it? To get that cash back. So yeah, down the gradient's always easy, no energy. Up the gradient always requires energy. Okay, in a 40% sucrose solution, this is such a common question. That's why I threw it in here. First off, even before we get to these answers over here, because we're talking about just one compartment, by the way. This is only one compartment. This is just a one solution. We're not talking about putting any kind of cell or a bag or comparing a different beaker. It is one beaker. That's it. One solution. So if I do something like this, let me, I guess I could try to draw this. If this represents a beaker real fast, okay? And that beaker is labeled what is it, 40%?
Yes.
If that beaker is labeled 40% sucrose, then that could be a test question. If you see a beaker and it's labeled 40% sucrose, here's the question. What is the solute in that solution?
The sucrose.
The sucrose, perfect. So the question was basically the same thing as the answer. If it's labeled 40% sucrose, the sucrose is the solute. That's what I'm trying to get across. Now, in this question here on the quiz, it asks, what is the percentage, right? What's the concentration of solute? And again, there would be the 40%. So here I should have added in my beaker that it was sucrose, of course. But I can't really fit that in there, so I'll just put that. But yeah, what about the solvent? Same compartment, so now we can equal up to 100. How do we do that?
It would be C.
There you go. You wouldn't believe every semester I do this, and without the coaching, of course, people get this wrong. And that's okay. I understand it completely. But here's the deal. I care so much, I don't want you to get it wrong. So study it in the right way. We're talking about one compartment, so therefore equals 100. And notice where there's less solute, there's what? More water. If this were an 80% sucrose solution, would there be less water?
Yes.
Yes, because then there would only be 20% water. So there, right there, you just confirmed that whole opposite logic thing. Where there's more solute, there's less water. Here, there's less solute, therefore there's more water. It's just playing the game off of 50-50, you know? If it's an inequality, it's going to be more or less something else, right? If something's less, something else is going to be more. Always, always. Okay, perfect. So I need to scroll down, so I need to clear out these answers. Everybody good so far?
Yes.
Wonderful, wonderful. Hopefully you fixed some possible snags in your learning of that. Okay, so for this next one, I'm actually going to bring in the iPad to hit this one correctly because I can't draw that much on that screen. So let me pull this up. Just give me a moment. Gotta love iPads. All right, let me drag this over real quick. Everybody seeing the whiteboard?
Yes.
Beautiful. Okay, so let me pull my quiz over this way just for a moment. Great, so I'm gonna have, on this beaker here and the cell being described. Whoops, wrong one. Okay, so here's our beaker. And this is what I would suggest to you. Anytime you see a picture like this or a question, if you've got scratch paper with you there on the test, sometimes it's better to just write it out, right? And make it on your own terms so you can understand what's going on. So we've got a cell.
I'm sorry, can you do that when you're taking the test in honor lock?
Yes, you can. The only deal is you got to show your scratch paper before the test, before you start on camera, front and back. And each time you use the sheet of paper to write on, you've got to show front and back. So it can be proven that you're using the same piece of paper and that you don't have anything else on it.
Okay.
Yeah. Okay, so here we go. Number 11 and 12, we're going to do on the same picture here. So a cell is placed, a cell filled with 78%. So let's get that going on. So here's our cell. What's it filled with?
78% sucrose.
78% sucrose, great. So let's go ahead and write that, 78%. And then it's placed in a solution of what?
54% sucrose solution.
Oh, my pen's not writing correctly. Hold on. Oh, let me switch pens. This one's not working good. I hate that. Okay. So right away, we see a gradient established. When I say that, it means that there's an inequality of the solute concentration. That's what creates a gradient, because it means that solute will want to move down its own gradient, and water is going to want to move down its own gradient, right? So we already know water chases the high solute, don't we? Because we've said that now, what, 8,000 times? Or 8,001, you tell me. Let this be 8,001. So where's the high solute?
In the cell.
In the cell, absolutely. So therefore, where's the water going to be driven towards?
The cell.
Inside the cell. That is great. So we got a good net flow of water chasing that high solute inside that cell. Now here's that trick question. Will water move in both directions?
Yes.
It actually wants to. It tries a little bit, right? Go in the opposite direction. But the net flow, the given flow, the gradient flow is like this inside the cell. How can we prove it? Well, we can do percentages if you want, if that helps. Sometimes that does help people. So let's do some percentages. What's left over? What's 54 from 100? So that would mean we got 46% water outside, right? If we had to do it that way. And what about inside the cell? 22. So you tell me, 46 to 22, does that sound like downhill?
Yes.
Heck yeah, we're going from an area of high concentration of water, which is 46, to an area of 22. Did water obey the rules for passive transport downhill? It sure did. But did a water obey the rule of osmolality, where high concentration of solute attracts water? Yes, it did. So ding, 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 we got two answers correct there. The net flow is coming in the bag, so the bag gains weight for number 11, or gains water, you should say. And then number 12 in this quiz shows you the net movement, which is inside. So on your actual test, it might show you a figure of the bag, in a beaker like this or a cell, whatever, and it's going to ask you, given the solution concentration, what is the net flow? So it's up to you to draw your own arrows, either in your head or on a scratch sheet of paper. But I'm just telling you, water is always going to be driven toward that high solute. So that's the easy way to do it. So if we take that same idea and run it over here in different terms, like we love to do, right? Let's take Let's take a red blood cell now, placed in what we're going to call a hypotonic solution. Right? Popular question. What does hypotonic mean? It means low solute, doesn't it? You tell me, the picture on the left, did the solution that the cell is sitting in, was it lower in solute concentration or higher? On the left.
Lower.
The solution was lower, it was 54. On the right, same exact scenario, just given in different terms. We're being told that the cell was placed in a hypotonic solution, which is the same exact thing that happened on the left. So automatically, if we know that the solution is hypo, given the game of opposites, what would be the concentration comparatively inside the cell on the right?
Hyper.
It would be hyper, wouldn't it? There you go. So in this scenario, the cell looks to be hyper. So where's the water going to go? It's going to chase the higher solute all over again. Because comparatively, if one compartment is hypo, automatically the other compartment has to be hyper. That's the done deal. It's a comparative term. You have to have one or the other. That's how it goes when you're comparing compartments. One compartment's going to be hypo, one compartment's going to be hyper. So the question told you hypo on the outside, so you assume that in this case, the cell is hyper, which tells you the water's going to go in. So that should answer some good questions there.
Now, are you doing, you were just doing 13, 14, and 15 to red blood cell?
I was just comparing how this numbers 11 and 12 is the same thing as talking about the ones we're about to do, 13, 14, and 15.
Okay, I got
Yeah, I was showing you how they could be similar, just using different terminology.
I'm confused about the cell. You said it's a hypertonic cell, but isn't it swelling?
It is swelling, yeah.
So is that not hypo?
No. You're mixing up how it swells. It's placed in a hypotonic solution around itself. It'd be like you, right? How tall are you?
5'2".
5'2". If you walk into a room of people that are 3'5", right? If you're 5'2" and you walk into another room of 10 people that are only 3 feet tall, are you tall in that room?
Yes.
Yes. If you walk into a room of people of 10 feet tall, are you short in that room?
Yes.
Yes. So whatever environment you're put in, you're going to be labeled differently.
Okay. So if it's a low solute for the hypo, then it would.
Yeah,
be the opposite.
it appears to be hyper when it's put in a hypo solution.
Okay.
Yeah.
Thank you.
So you're welcome. Everything is comparative. That's what I was trying to get across. But that's great that you ironed that out. That's wonderful. I love to hear that. Yeah, so that cell, that red blood cell on the right, is a normal cell. Nothing wrong with it. But when it's placed in a different environment, which is the solution that we're talking about, hypo or hyper, that's when it's going to react differently. So therefore, it is labeled differently in a different environment, basically. So let's squeeze those in real fast. So everybody good with 11 and 12? Okay, so let's start over. Let's get all that straight, because I find this red blood cell stuff, this same question, oh my God, back in college, holy cow, I had this back in 1988, right? This when I was in college. Yeah, and I hated it so badly that I would actually physically turn the page and probably say a few cuss words because I couldn't stand it so bad. Because it never made sense in class. Either that or I wasn't paying attention. I was just looking at the girls in class. You know how it goes. That's how it is in young college days. Especially at Ole Miss, all the girls, that's truthfully why I even went to Ole Miss. Isn't that stupid? The recruiter came to my high school, and that recruiter was so cute. I thought, hey, I'm not going to go to LSU, even though all my rest of my brothers and sisters did. I'm going to go to Ole Miss because that girl was cute. How stupid is that, right? That's the mind of a guy, I guess, at least at that age. Well, here you go. We got two beakers. I'm just going to do the hypo and the hyper because the ISO just means same solute. therefore the same water movement. If you didn't catch that, it's the solute concentration that dictates the water movement. That's it. Solute rules the game in this case. So if it's iso, which means same solute concentration inside and outside the cell, then that water movement has to be equal, right? Because it follows the solute just equally. So we'll just leave that one alone. There's no net flow there in iso. No net flow, just equal water movement both in and out. So let's do the hypo and the hyper examples. On the beaker on the left, we're told that it's put in a hypertonic solution. So let me just write
that would be more solutes outside of the cell.
Yeah, we're talking about that solution, right?
Okay.
And on the right,
more solutes in the solution.
Yes.
Than in the cell. And,
yes.
And that's the causes the cell to shrink.
Yeah, that environment is going to cause that cell to shrink or swell. So here's our examples describing the solution, okay? We haven't said anything about the cell yet. This is just the solution that's being put in. So just like you mentioned, let's translate what hypertonic means. Hyper means increased, right? Tonic means solute. So all we know right now is that solution, never mind the cell right now, we're talking about the solution, has more solute, right? It's high in solute, which means it's above 50%. If you wanted to assign a percentage, it doesn't matter though. This solution on the right has a lower solute in it. Okay, so if we wanted to play the opposite game, which I love to do because there's opposites all through A&P, man, I'm telling you, if you can recognize opposite logic, you're going to be clean sailing through a lot of principles in A&P. So let's talk opposite logic. When you get high solute, what do you get with water in that same compartment? Low water, don't you? In that same compartment, wherever there's high solute, there's less water. We said that earlier. What about low solute? What's the deal with water there?
Higher.
It's higher concentration. That's the game of opposites. In a single compartment, we proved it with that 40-60, 60-40 on number 10, didn't we? So this is playing the game of opposites. If it's a hypertonic solution, you have high solute, which means less water in that solution. And then hypo is just the flip of that. Now let's take our red blood cell, make it real. We put a red blood cell in these two solutions. There's nothing wrong with the red blood cell. Its tonic, its tonicity is just fine. But when placed in two different environments, it's going to react two different ways. So comparatively, if we're saying that it's high solute on the left in the solution, comparatively, it would be what in the cell on the left?
Low solute.
Yep, comparatively, it would be low solute. And what about on the right?
High solute.
Comparatively, it would be high solute in this scenario only, by the way. So this is just a more complicated way of looking at it, but sometimes people need the details. What we're really concerned with is, even before we drew the red blood cell, where is the high solute on the left? It's in the solution. Therefore, the water will chase the high solute. Always, always, always. Okay? And what about on the right? Where's the high solute?
In the cell.
In the cell, comparatively speaking. So, did water move down its own gradient?
Yes.
Yes, it did, because let's take the example on the left. It started off low right on the outside, but water was higher on the inside, wasn't it? Because it was less solute. Remember the game of opposites. If you can know the game of opposites, and if you can write this down, by the way, on your own, then you own this material enough to teach it next semester. I'll give you my job.
So,
water moved from high to low, didn't it? It moved in both scenarios from high to low, didn't it? Otherwise known as chasing the higher solute, or being attracted to the high solute, is how it's mentioned in the book. Okay, go ahead.
I'm just wondering, on that hypertonic, would you think of that as like net flow out?
Oh yeah, absolutely. Yeah, we're definitely going to mention that.
Okay, okay. I'm
Yeah,
trying
so
to,
when presented with these type of questions, the first thing they're probably going to ask you is if the cell is going to shrink or swell. Will it crenate or hemolysize, right? So the one on the left, in a hypertonic solution, it shrinks, which we call crenate with an N, C-R-E-N-E-A-N-A-T-E. And on the right, since it swells, we call that hemolysis. So yeah, let me caution you, whenever you get a red blood cell example, you're going to want to use that crenate and hemolysis terminology because that's what they're looking for. So all I did by making it look complicated here is to prove to you that we obeyed two simple rules. Well, three simple rules. The first rule being its passive transport. Water always moves down its gradient. And by playing the game of opposites, notice all I started off with was high solute on the left. Then playing the game of opposites, I'm able to fill out the other three. Can y'all see that? If you're starting off with a high solute solution, hypertonic, then whenever you enter that red blood cell, you can fill out all the other tonicities based on opposite logic. Because in the extracellular compartment, it's low water. On the intracellular compartment, it would have to be high water because it's all opposite. And it would have to be low solute because it's all opposite. So try that. Try this on your own. Take a snapshot of this slide. And then at some time later in the day today, I would do it today before it slips out of your head. Try drawing these two pictures and filling out everything we just did. See if you can get it. And once you get it, it's yours forever. You don't have to study it anymore. Consider it done because it all makes sense to you, hopefully. So water did move down its own gradient from high to low. Notice that in the blue. Otherwise known as being attracted toward the high solute. So for me, the quickest way to get this is to look for the high solute, and that's where the water's heading. See you later. That's it, right? If it's hypertonic, that's where the water's going. If it's hypotonic, it means the water's going in the opposite direction. That's the 60-second or the 10-second way of learning this, to be honest. All right, any questions on that? So to being asked net flow, the net flow is out of the cell here in hypertonic solution, and net flow in the cell in a hypotonic solution. You could say equal net flow for ISO. I think that's what the book says. I don't know what's wrong with that author. There's actually no net flow because it's when you say net, you mean a favored direction. So to me, there's no net flow in isotonic because it's equal movement of water. Equal movement of water in isotonic because it's equal solute. That's what iso means, is same solute. All right, guys, so we'll leave this and go to our new stuff unless there's any lingering questions. We're about an hour in, that's fine, because the PowerPoint, there's not much to talk about there, just looking at some cell components. But other than that, we have a worksheet to complete that'll take us about 30 minutes as a class. We'll break out in groups for about 15, 20 minutes, and then we'll come back and spend another 15 minutes going over it, that same worksheet, since there's no quiz on it. But yeah, we'll be here till 2 talking together.
can you tell me, I need to print out the worksheet and that's in course content?
Yeah, I'm about to pull it up and show you where it is. Yeah.
Okay.
Okay, so yeah, definitely take a snapshot of this and practice it on your own. Even though it only represents possibly 2 questions on the test, it helps overall because it makes sure it confirms that you know how osmosis really works. The nature of osmosis and the word osmolality to go with it means that solute attracts water. So yeah, we keep talking about all this water movement, but water does not do a darn thing unless solute is pulling it. That's why nothing really happens fancy in isotonic. This water is naturally moving in both directions anyway. So iso is just confirmation of that. It's just equal movement in both directions. But yeah, it's solute that drives the game. That's what pulls the water. That's what osmolality means, by the way, is solute concentration pulling water or attracting water. And that's all osmosis is. It's attracted to high solute. Down the gradient, no energy required. It's just a natural attraction. It's going to want to dilute that solute every bit it can. That's why you get high blood pressure if you eat salt too much in your diet, because that salt circulates in the blood, draws water into your blood, raises your blood pressure. As simple as that. Okie doke. Now let's look at this new the new worksheet we've got for lab today.
Oh,
So let me pull this over. So yeah, go to our course content. And then we're going to be looking at lab 4, of course. Whoops, wrong one. Let me collapse that out of the way. Lab 4, here we go. Lab worksheet right there. If you're clicking on lab 4 instructions, like I'm urging everyone to do, because I'm getting some emails of people missing some data, some info, which is a common thing every semester. But always look through the instructions first and you're given everything. I put this link there recently to help out. That's another link to that same worksheet. So when we open up that worksheet, this is what we get. We get in lab what we do when we're there in lab, I should say, on campus. We have 4 or 5 stations that we have all the students break out to. And at each station, there's a model, or a question of some kind. Here at station A, there's a model of this little doodad, the cell membrane. So it has some questions based on that, right? Then you move to another station. There's a question about aquaporins. That's cool with Jasmine. She's pregnant. And then another cystic fibrosis case study. So a number of case studies in this lab today that we'll go over. But they really want you to try to figure these out based on what you already know. If you can't figure them out, no problem. We're going to go over it. But try your best to figure it out as a group. I'm going to put you guys in groups again of 4 or 5. And then you're just going to identify cell components, these organelles, and their function right here. So again, you're doing this as a group, more cell components, and then some other random stuff down here. I'm not so concerned about this stuff. So go as far as number 32, to be honest, because that's the relevant information for our test. Past 32 starts talking about info that we're going to get into next week in lab. So my view, because I'm just looking out for you guys. Yes, this is part of the lab worksheet, but truth be told, I'm fine with you guys stopping at 32 because that's what's relevant for your test. I think a better focus is what's needed right now. I don't think we need anything new, do we? I think you got enough. So stop at 32. If you want to do these, you can, but otherwise, I mean, they're fun, I guess, but you can stop at 32. So everybody print this out. There's a print option right here at the bottom, right there. I would recommend not downloading right now because it's just going to give you a zip file and all that mess. Just print it out here. Or if you cannot print it, you're just going to have to write your answers on a separate sheet of paper. But yeah, okay, so we're about to break out in different rooms to do this worksheet. But first, I want to actually give you a little something on the PowerPoint. It's not much, just a little bit to help you along, because a lot of these answers, I'm Mr. Helper today. I don't know what else to say. I try to help. A lot of these answers are going to be in this PowerPoint. So let me take maybe 10 minutes of your time here to quickly go through some things that will help you on that worksheet. And hopefully that'll make it go faster, so maybe we can get out sooner. Okay, so looking at our beautiful cell, of course, we know our cell membrane has all of its characteristics that we've talked about, phospholipid bilayer. All those membrane proteins are just not shown in this picture, of course. They're focusing here on the inside, the cytoplasm. So let's distinguish cytosol from cytoplasm. Sometimes they're used interchangeably, but by definition in the text, the cytoplasm is the area inside the cell. You could say it's the intracellular compartment, but the fluid, the water, and the random amount of proteins and other things floating around, that is the cytosol. So the fluid is the cytosol, the area inside the cell is the cytoplasm. So then we got all of our friends in here, right? Gonna be the same old friends we see in every cell. There may be a cell that does not have a nucleus like the red blood cell, or the ER may be different, like in muscle cells. It's specifically a little bit different there, but pretty much the same old thing in every single cell. We're going to have cytoskeletal fibers, these intermediate filaments, proteins. By the way, would they be fibrous or globular? What do you think?
Fibrous.
There you go, fibrous, right? In this case, they'll be a secondary because they have a little bit of a coil to them, right? But no matter, it's just a protein. That's a cytoskeletal protein fiber that gives the shape and endurance to the cell, you could say. Then we've got some other friends like the mitochondria for ATP production. That mitochondria is going to be what's called the powerhouse. It takes one molecule of glucose among thousands and converts that one molecule of glucose through several, complicated processes and gives us up to 38 molecules of ATP. So isn't that pretty cool? Then we got lysosomes acting as the digestive system to the cell. They have enzymes inside that break down all kinds of toxins and other things that come inside the cell. Then we have the nucleus, of course. Probably the biggest of all the organelles, centrally placed usually. Some cells have more than one nucleus, like muscle cells. They require a lot of protein synthesis to keep them happy, so they're going to have more genetic information to make all that happen, at least in one spot, right, in that cell. So some cells can be multinucleated is what I'm trying to say. Anyway, okay, so the outer membrane of this nucleus is called the nuclear envelope, or membrane, but its envelope is another word. It has these pores in it, which we'll talk about in a second. Inside the nucleus is called the nucleoplasm. Just like this is cytoplasm, this will be called nucleoplasm. So all this generic faded purple here is going to be where your DNA is. It just doesn't show it. So we're going to know that DNA as chromatin. So in the nucleoplasm is the chromatin, which is the condensed form of DNA, all coiled up. Chromatin and DNA are basically the same thing. We just say chromatin because it's coiled up with some binding proteins. And then at the center of the nucleus is the nucleolus. You can see it's kind of bulging. It has some ribosomes inside that are just itching to get out. So this is where ribosomes are formed, in the nucleolus. So on the test, if you see the word nucleolus next to the word nucleus, you better look for that extra O in the nucleolus, because you don't want to use the wrong word if you're labeling something. The nucleolus is inside the nucleus, right? And that's what produces the ribosomes that we're going to see in just a bit. All these little red dots, these are the ribosomes. How do they come out? They fit through the pores. That's how they come out. Now, these ribosomes could float around on their own, which are called free ribosomes over here, just floating around fancy free. Or most of them are going to be bound to what we call this ER, endoplasmic reticulum. Endo means inside, plasma means cytoplasm, reticulum means a folding network. So notice all this ER is just folds of this material here, also found right here. And now they'll pay attention to the colors so much because the colors just, don't really mean anything. They just help you identify. But yeah, where there's ribosomes on the ER, we call that rough ER. Where there's no ribosomes, we call it smooth. The smooth comes off of the rough, so it's the same ER. It's just that this portion of it is rough because it has ribosomes. This portion does not have ribosomes. It's called smooth. Then the ER wants to communicate also with this other one, the Golgi. Notice how the Golgi could look similar to the ER itself. The difference is you'll see these little bulges come off the Golgi. These are vesicles that we saw in lab for exocytosis, where we carry things out and push them out of the membrane. Well, it just so happens that the Golgi packages all the newly produced proteins on the ER, this rough ER, packages them up and then puts them out of the cell, either embeds them in the membrane or pushes them out, if it's a hormone or enzyme, something like that. So yeah, let's talk about that for a second. Rough ER is big on protein synthesis. That's its big job there. And since it's such a big organelle, it spans really the width of the cell. It'll transport those proteins also. So the nucleus, known as the brain of the cell, is going to have the info, the genetic information to build the protein inside. Then we copy that information, bring it to the ER to build the protein. Now we give that protein, we transport it to the Golgi, I should say, also. So production and transport on the ER here, the rough. Give it to the Golgi to package it in a little vesicle, and then that vesicle exits the cell. So it's in the PowerPoint coming up, that little sequence there, but it's worth mentioning again. If you're asked what the passage of a protein is inside the cell, 4 places. We start off in the nucleus, then we go to the rough ER, then we go to the Golgi, then we go to the cell membrane. Those are your 4 places for the passage of a protein. And it's listed in the PowerPoint coming up. But I'd just like to use this slide as an intro, just to talk about things before we see words. Sometimes people are distracted by seeing all the words. So I just wanted to use the picture first. All right, so let's do some words. You can skip through that. Here's all the organelles we mentioned. Peroxisomes are going to look similar to a lysosome. They just detoxify a little more than lysosomes do. 3 regions of the cell that are primary right here. That's usually a popular test question. Of all the places in a cell, what's the most primary regions? There you go. Now looking at the cell membrane, we all know well about that. It's known as a mosaic because we've got all these things embedded. These are all the protein uses of the membrane proteins. We've done all that in lecture. So this is more for lecture over here. We've already done that. It's just a review slide. First up on your worksheet today is getting to know the structure of that membrane though. So let's review that. We definitely have that polar phosphate head that makes a hydrophilic region. You have to know all that right there. That's going to help you with your first question, your first station. 2 nonpolar fatty acid tails giving you a hydrophobic region. So get all those words together. And then this picture here, you might want to snapshot that, label it in your head. What is polar? What's nonpolar? What's hydrophilic? What's hydrophobic? What is a phosphate head? What's the fatty acid tail? All these things need to be labeled. So here's the labeled version of it. Can you draw this, which is one of your activities in the worksheet? Can you draw this and label it? Right? So these are all your tails, your fatty acid tails, right? Would they be hydrophobic or philic?
Phobic.
Phobic, right. And these are all the phosphate heads? Phobic or philic?
Philic.
Philic, right. Are these polar or nonpolar, the heads?
Polar.
Polar. And the tails would be?
Non-polar.
Nonpolar. Excellent, excellent, excellent. That's all you need to know. That's it. Beautiful. Practice that. Easy stuff. No reason to get it wrong on the test at all. Not a reason. Just don't get it mixed up. Now to make things interesting, I threw this slide in there. So you may not have it unless you've downloaded the updated PowerPoint. But this is also used in our lab. So this could, I mean, I have no idea, but this could very well show up on the test. Well, and I know in lab, if we're on campus, we always have this model out. You know, of course it's out. It's what's the major thing we study here in this lab. So yeah, I would know this model really well in reference to the same things we just learned here. Nonpolar tails, hydrophobic region, polar heads, hydrophilic region. Now what's this fancy thing in the middle? That is just a general membrane protein. We don't know if it's for active or passive. It really doesn't matter. We don't know if it's a carrier or a channel. It really doesn't matter. All we know is that it's a membrane protein. Now here's a question. Besides just labeling that as a membrane protein, what if it was labeled and it asked, would this be used for simple or facilitated diffusion? What would the answer be?
Facilitated.
Facilitated.
There you go. There's your topic right there. It could be used for facilitated transport or active transport. So I could say it again. Would this be used for simple diffusion or active transport?
Active transport.
There you go. Now you're getting to be flexible with the material. That's what it takes to do good at these tests. You get the generals, core content, and you know how to conversate with it. That's what's important. Great, 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 great. And then of course that's just recognized as the intracellular compartment versus the outside. So you may be asked something simple like that. Who wants to miss that? Hopefully you don't. Okay, great. Great, great, great. So another fancy slide here. I gave you this one in the handout just the other day, a couple of days ago in the email to have you practice labeling all this. I gave you a blank one and a labeled one, right, in that handout that I emailed everyone. So it's just another way of practicing. The centriole, I think I missed in that last picture. It's important with mitotic direction. So it uses these microtubules coming out in the yellow here to basically push and align those chromosomes to the center and then pull them apart when we do mitosis next chapter. That's what organizes. So it's like a conductor, like a conductor in a symphony, truthfully. It conducts these chromosomes to do what they need to do for mitosis. All right, excellent. So here's that passage I was talking about, those 4 areas of the cell. That's going to show up on that worksheet also. See how nice I am? I don't know. Maybe I shouldn't be so nice.
This is on the lab 4 worksheet.
Yeah, all these are gonna speed you up through that worksheet. Absolutely.
Okay, so it's not in this packet worksheet.
Now, which one are you talking about?
Just what you've been, all of these that you're showing, they're where?
The PowerPoint is going to help you with the Lab 4 worksheet that we're about to do.
Right, where's the PowerPoint?
Oh, the PowerPoint is the same one we've been using. That's listed under Dr. Sliman PowerPoints for lab on the left in course content.
Yeah, and I've got that out, but I'm just not finding. I've got cytoskeletal fibers, mitochondria, where it's just, really breaking it down, but I don't see.
You're further down. I haven't gotten there yet.
Am I further into the packet? Okay.
Yeah. Just go a little bit earlier. Yeah.
All right.
Yeah, these are just the first like 10 slides that I'm looking at. So there is all your summary information. So when you get to those stations where you're asked the function of these organelles, there's your go-to right there. Make it real nice and easy. There's all the functions. So this is high school stuff. Nothing's changed. All the same thing. You just got to cram it in your brain one more time. Okay, so here's where you were, Cynthia. All these things right here, break each one down.
Yes.
And then we'll talk about those case studies afterwards. So that's all I wanted to bring to you right now. I'm gonna break you out in rooms. And so you guys can work on this worksheet right here. Again, it's under the cell biology lab. Just click on it and there you go. It has, you can do stations A, B, and C. You don't have to worry about station D and E. Don't worry about that. Go down to number 32 is really what we need to do. And then we'll go over it. So I'm gonna break you out for about 20 minutes or so. It should be really quicker if you're using the PowerPoint as your guide. And then we'll come together and go over it all. And then we'll be done, okay? So just accept the pop-up that you get for your breakout room. And by the way, did I answer everybody's questions for that quiz earlier? Hoping y'all did pretty good. Okay. Okay, basically the same rooms as we did last time. So here you go. I'll see you in about 20 minutes. And talk together, by the way. Work through this together as a group, if you could. Okay, so we're all back. There we go. Wonderful. Well, we'll be working through this worksheet again with the answers. And since there is no quiz on this to be completed in eLearn, I thought it would be great for us to take the time today like we've done. So I appreciate your patience with doing that again. It's something we normally do in lab anyway, so we might as well make it real and complete all this here, because I find, just like I did yesterday in lab, it is a huge benefit to finally be confronted with these questions that probably are worded differently than how I would put it, but that's a good thing. That means that you're being challenged with the same information, but in different words, which is exactly the experience you need for the test, because some of these same things off this worksheet could show up in a different form maybe, but certainly on the test. So like I said before, if you didn't catch that in one of my earlier lectures today, definitely for each of your 4 labs, go through any worksheet, any of them that you would have done. I'm talking about the eLearn application handouts that were there for download, and anything you took a quiz on, of course, but those downloaded application lab handouts are your best study guide when it comes to the lab portion of this test. Okay, so let's get to it. Let's go through each one. Let me get mine up here. Just a second here. Okay, so looking at number 1 and number 2, let me see if I can, well, that's about the most I can do. That's fine. That same model that we saw earlier, what do the white balls represent?
The phosphate groups.
Yeah, the polar phosphate heads that are hydrophilic. So you really want all 4 words, definitely. You want polar, hydrophilic, phosphate head to be clear, because you never know in which direction that test question will be asked. So all 4 words will be appropriate here. One more time: polar, hydrophilic, phosphate head. And then for the tails, you would want nonpolar, hydrophobic, fatty acid tails. That's covering everything, to be clear. Let me move my computer here just a bit. Okay, great. What about this blue thing? What does that represent? Which one of these?
Membrane protein.
It's a protein, for sure, a protein, absolutely. And then hopefully you sketched a little something down here, roughly the same thing over here, and you labeled it, the phosphate heads, the fatty acid tails, which area is hydrophilic, hydrophobic, polar, nonpolar, and you get that all from numbers 1 and 2. So yeah, I'm sure on the test you'll likely see some kind of a cell membrane that you're going to have to label, whether it's philic or phobic, polar, nonpolar, fatty acid, or phosphate. So get all that straight for sure. And the function, selectively permeable. Well, in general, the word permeable means to pass through, but we say the word selectively permeable because it means some things pass through easier than others. That's the best way to put it. Certain substances pass through easier than others. And to go a little further with that, if you look at this picture again, comparing the red to the white, what do you see more of? More red or more white? Like, what catches your eye? Certainly there's more red, right? So that thickness of that cell membrane is all nonpolar. Therefore, the cell membrane will behave in a nonpolar fashion, which is going to dictate all of our things coming up 5 through 11. So when you see the word selectively permeable membrane above that term, to make it easy, I would just go ahead and write nonpolar. Because yeah, the phosphate heads are polar, but for the purposes of us understanding the greater physiology going on, think of the membrane as a nonpolar membrane, because 90% of the real estate of that membrane is nonpolar. Therefore, that's how it's going to behave. Molecules will slip right by those phosphate heads because in real physiology, there's gaps between them. They're not really touching. So lipid soluble molecules are going to go right past those phosphate heads. They're just going to wave. They're going to say, yeah, no biggie. I'm headed for the nonpolar region. Headed in or out. So that membrane is nonpolar. Just put it that way. That makes things a whole lot easier. Now, to be specific, it is both polar and nonpolar because we got polar phosphate heads. But I'm telling you, for the physiology of it, for answers 5 through 11, think of the membrane as strictly nonpolar. That's the best way to go for it. So based on that knowledge, using simple or facilitated, simple meaning lipids, right, or fat soluble molecules, and then facilitated for water soluble molecules or ions, right, that have a charge. So let's go for it. Glucose, it's polar, therefore which will it use, simple or facilitated?
Simple.
Nope, because simple is for nonpolar.
Facilitated.
There you go. And that's good to get it wrong, because now you know forevermore. Glucose is polar, therefore it has to use facilitated, because anything polar has to be facilitated, which means it needs help. And that membrane protein is what we need. Or in this case, it's a carrier, isn't it, for glucose? To look in the PowerPoint, it's right there. For nonpolar O2CO2,
Simple.
this is simple. We want these to have free access through the cell membrane because guess what? All cells need to be oxygenated. They need to pull in that oxygen real fast. We don't want to wait for no protein. We want to go right through. So that's why these gases go right through. And CO2 is a waste product, so we need to get rid of it real fast. So we get rid of it real fast right through the membrane. That's what we're going to see in AMP2 when we do the respiratory system. We'll see the communication between the alveolus and the lung, the air sac, versus the capillary that's right next to it. It's going to exchange O2, CO2 in opposite directions, right? Fresh O2 from the lung and the cell that's used up, right? The blood cell in this case, gives its CO2 back to the lung so you can breathe out. So it's that opposite exchange again. And they go right through the cell membrane. If you didn't realize this, when we're talking blood vessels, that blood vessel wall is not solid. It goes right on through. There's plenty of holes in it. They're small. And those gas molecules go right on through. Same with the cells that they oxygenate. When you get a cell that's oxygenated, that oxygen's going to come right on through that cell membrane. Thank goodness for it. Hydrophilic, is it simple or facilitated?
Facilitated.
Facilitated, yes. Hydrophobic.
Simple.
Simple. Very good. Cation, anion.
Facilitated.
Perfect. Facilitated, because these have a charge. Anything polar or anything with a charge is straight from the PowerPoint. Go check it. Will be facilitated. Steroid hormone.
Simple.
Simple, right? Because simply steroids, lipids, and gases like O2, CO2 that are nonpolar, yeah, they'll be simple. And fat soluble vitamin like vitamin E.
Simple also.
Simple also because it's fat soluble. Those fat soluble vitamins, if you don't have them in your memory yet, it's A, D, E, and K. Let me jump here real fast. I wanted to do this so we have it on the recording because not every class, I think, got this. So I'm going to pull up my whiteboard once more. And I want, in this area here on the right, whatever I put on the whiteboard, I'd like for you to go ahead and add that to your sheet. And it'll greatly explain how to work through 5 through 11 really quickly. And yes, I could have put it there before, but I wanted to see you guys wiggle a little bit. That's how you learn. So squirming and frustrating, all that kind of stuff, it's all helpful. Well, let's do this. I'm very big on this when I'm on campus. I miss having my whiteboard, I can tell you that, because I put lots of things on the whiteboard. Well, let's do it this way. Let's make 2 columns. Polar versus nonpolar. And we're talking word association, okay? So just write down the word association for each polar and nonpolar. First
Dr.
off,
Sliman,
yeah, go ahead.
are we supposed to be able to see your screen?
Oh, can you not see it?
No, sir.
Oh, I'm so sorry about that. Yep, that's my fault. 1 second. Thank you for letting me know. I thought that I'd already shared it.
No problem.
Thank you, thank you, thank you. Yeah. So for that recording, let me go back real fast just for the sake of the recording. Where we were working is these first few questions here. 1, 2, 3. We sketched the membrane for number 3. We talked about the reason why it's selectively permeable for number 4. And we completed 5 through 11 just now. And we'll go through those answers once more for the recording for confirmation of that. Thank you very much. Okay, so now we're making a side-by-side. Thank you. Polar versus non-polar. So I want you to write what I'm writing here in this area right there, to the right of 5 and 11. Make yourself a side-by-side. We want to know that anything polar is going to be known as water soluble. Let me write that again. It's in a little bit thicker than I wanted. Which makes non-polar what? Water insoluble. Let's play the game of opposites already. See, I started off with 1 and I can do 3 more. I start off with water soluble, then that means non-polar is water insoluble. So what is non-polar then? Lipid soluble, right? Which means polar is what? Lipid insoluble. Play the opposites. So with polar, what word goes with polar? Hydrophilic or hydrophobic? Hydrophilic. And then for nonpolar?
Hydrophobic.
Hydrophobic. Great. As far as diffusion goes, which one's polar, facilitated or simple? Facilitated. And nonpolar would represent simple diffusion. Solutes moving simply right through the cell membrane, because what's the cell membrane? Is it polar or nonpolar largely? It's largely nonpolar. That's why nonpolar compounds simply move right through. So I see that that little watermark is covering it up. Sorry about that. That says simple underneath hydrophobic. I don't know why it says trial. I've purchased this thing like 3 times. It keeps telling me it's a trial. I need to talk to them. So there you go. The only thing left that I don't have room to write would be the examples. So under polar, for facilitated, you could write, I don't have any room, but you could include this. You can include sugars, amino acids, and ions for facilitated movement. Sugars, amino acids, and ions. And of course, it goes without saying, water itself could be included in that list. So sugars, amino acids, ions for polar. Under the nonpolar column, you could write lipids, steroids, and O2, CO2 gases. Because not all gases are nonpolar, to be honest. But O2, CO2, and methane, those are all nonpolar. So we mainly talk about O2, CO2. You can include that. And if you want to jot down fat-soluble vitamins, you can put that there too. So there's your quick way. There's your 10-minute study session right there. Memorize this chart. Memorize the word association, I should say, or recognize it even more so than memorize. And then test yourself. Can you write this on a blank sheet of paper? Do it until you can. That's my best tip for you.
I have a question.
Yeah, go ahead.
On number 9, it's the cation or the anion, and it says that, or we said it was the facilitated diffusion,
Yes.
but polar ions are polar.
Yeah, but ions have a charge, don't they?
Yes.
Yes. And since they have a charge, and polar compounds have a charge, they mix well together.
Okay.
So you're right, ions are not polar. That's a great distinction, but they do have a charge. So they mix well with water because water has a charge. It has both charges.
Okay.
Yeah, so anything with anything that has polarity, like a sugar or an amino acid, or anything that has a charge in general, will be under the polar, or excuse me, under the water soluble heading, which would be facilitated. Okay, great. Moving on. For number 12, we look at the aquaporins, of course, and we talk about water being moving through the membrane, either of these two methods. Well, the preferred method is always facilitated. Osmosis is just a kind of facilitated diffusion. The book may not say that very clearly, but since we're using a carrier, the aquaporin, anything with a carrier, in this case, if it's passive transport, is going to be facilitated diffusion. So osmosis falls under that category. It does move under simple, but very accidentally, not very reliably at all. So the answer there is definitely facilitated. And the reason why is because it has its own dedicated channel right here. It has its own dedicated channel. Okay. So yeah, they're found in high numbers. Yeah, that's fine. We're getting into the difference of active and facilitated. That's a good question. I'd like for you to also write something out here on the right side of 14. Let's do that first, actually. How is active transport and facilitated diffusion alike? How are they alike? And then we'll tackle how they're different over here. They are alike in the way that they both use membrane proteins. So in the textbook, let's make it real, all right? I like to link up things. In the textbook, they call this carrier-mediated transport. And if you look in the PowerPoint, I've even got a slide on it. One of the types of carrier-mediated transport is facilitated diffusion. The other type is active. Why? Because they both use a protein. So that's how they are alike. They both use a membrane protein, which is in loose terms called carrier-mediated. Now, how are they different is where one uses ATP, the other one does not. So which one uses ATP in number 14?
Active.
Active, right. So how are they different? One uses energy, which is active. The other one does not, which is facilitated. I love to try to cover all bases. That's one thing bothers me about the book. They don't really, they lead you in a certain direction, and then another page they refer to it differently. There's no continuity sometimes in the textbook. Okay, so for this case study here with Jasmine, she's pregnant. To make this long story short, when you're pregnant, the job of the body is to take care of that baby that's being developed, being made in the body. So it's less concerned. The body overall is less concerned, if you want to consider it its own separate machine, less concerned about the mother, very concerned about that newly developed baby that's going on. So the body is going to want to hold on to water as much as possible, because guess what? What's happening when you develop a baby? Proteins are being developed left and right and sideways, front and back, all over the place. You need tons of protein development to build a child. And you want proteins to live in a good environment, which is going to be a good hydrated environment. So women will naturally reabsorb and hold more water when they're pregnant, which means they're going to have more aquaporins. So whenever we have the kidneys compared to a blood vessel inside the kidney, here's the kidney tubule. That's a tube that actually is going to be concentrating the urine. So let me go back here for a second. So whatever stays in the tubule is going to become urine. And yes, I'm being corny and drawing a yellow arrow for urine being formed here in the kidney tubule. But guess what? All the water that's put into the kidney, coming from the blood, by the way, we're just filtering the blood to make urine. You naturally are going to have some of that water being reabsorbed naturally. Right? Whoops, wrong color. So some of that water is naturally reabsorbed out of that urine that's being formed, right? So it goes back in the bloodstream. Thank God for that. That's what keeps your blood pressure up. That's why you can't mess with the kidneys. Kidney problems are life-threatening, just like the heart. So that's in a normal case. A little bit of aquaporins here or there. I can draw a few if that helps. Here's an aquaporin channel. There's an aquaporin channel. There's an aquaporin channel for that water to pass through. But when you're pregnant, guess what? You got buku water channels. The body knows to produce more aquaporins because why? It needs to reabsorb a whole lot more water when you're pregnant. So that's what's happening here with Jasmine. She's reabsorbing more water. So the picture clearly indicates where are there more arrows? Over here, right? More water movement on the right. Which diagram represents Jasmine? She's pregnant. So yes, this one's Jasmine on the right, more aquaporins. Any questions on that one? Okay. Great, great, great. I'm gonna clear that out unless y'all want to take a picture of it, but that's what you should have drawn. Some of this is coming so much into AMP2, it's difficult to really find it in your brain. But really what they wanted you to know is the more water movement, the more aquaporins have to be there. Okay, great. Moving on. For this next case study with Jeffrey with cystic fibrosis, it's describing a functional chloride pump where there's no pathology would mean that you would clearly just be pumping out that chloride ion. Whoops. Let me just give a color here. The chloride pump will be pumping chloride ions out, right? In a natural state, in a healthy state. So that means chloride is being pushed out of the cell. Well, what likes to hang out with chloride? How about some sodium? So sodium's going to want to follow the chloride. And yeah, it's going to have its own little channel. So we can draw like a tiny little channel there. That's fine, if that helps. So chloride goes first, then sodium follows. Now you've created a hypertonic environment, didn't you, on the outside of the cell? So what's going to follow that? What's going to chase that hypertonic environment?
Water.
There you go. Water is going to use its own channel, the aquaporin, to chase those solutes out, right? So that's what's happening in a normal state. It's going to dilute the mucus to keep that mucus flowing. In a pathological state with cystic fibrosis, the chloride pumps, their proteins, of course, they're not made properly or they're dysfunctional. With cystic fibrosis being a genetic disorder, the proteins do not assemble correctly or the instructions for the protein are not there in the DNA. So what happens is there's no chloride movement, therefore there's no sodium movement, therefore there's no water movement. So this extracellular compartment becomes thickened. The mucus is thicker, which paralyzes the cilia. Then you don't get that regular movement of the mucus through the respiratory tract, and you get these mucus plugs, which is very dangerous in the bronchi. So cystic fibrosis patients have a limited lifespan because of that. I mean, there are treatments, of course, but at least at this point, you can't fix the genetic problem going on with the chloride pumps. But that's the deal with that. So if we look at the pictures, I need to clear this out to scroll. At the questions themselves. So we've got, why does sodium ion follow the chloride? Opposite charges, that's what it's all about. It follows the opposite charge. Sodium, which is positive, follows the chloride, the negative. Number 18, well, moving through these ions, will it change the solute concentration? Will it make the extracellular fluid hypo or hyper?
Hypo.
It moved solute outside of the cell, right? The chloride and the sodium?
Yes.
Which made the outside hypertonic, because we move solute in that direction on the outside. So it's asking about the extracellular compartment, which would be hypertonic. So then number 19, playing off of that, in which way would the water move? It would move out of the cell towards that hypertonic extracellular fluid. And that's what we want in the normal state, diluting that mucus. That's the normal state. So on the right is the pathological state, on the left is the normal state. And everything you need out of these case studies is going to be wrapped up in the PowerPoint. We'll go through those slides. They'll just take a minute. But real quick on these, there's not much time I can really spend on these, just to save you the 10 minutes of agony of more talking. But were you able to label these successfully? I can run through them real fast. 20 is the nucleus. 21, it can be a little tricky the way it's labeled there, because it kind of looks like the nucleoplasm, to be honest. But I think that what they were trying to tell you is the nucleolus, because that's where it's sticking. You just can't tell in this 2D picture. But this would be the nucleoplasm right here, which would be the DNA. So hopefully it's better labeled on the tests. Nucleolus right there, nucleoplasm here, where the chromatin DNA is. 23 is the rough ER. 24 is smooth ER, because there's no ribosomes on it. 25 is your Golgi. 22 is ribosomes, specifically called free ribosomes, since they're not bound to the ER. They're right there floating around, fancy free. Okay, so that's that one. You can write down those functions from the PowerPoint. Here, right here, points to our passage of protein slide from the PowerPoint, if you can remember that. So we saw very clearly, I think on this slide, I can move it over real fast. Right there. We start nucleus, proteins in the nucleus, to the rough ER, the Golgi, and the cell membrane. That's where that info is. So here we go. Genetic instructions, underline that keyword there. Where are they found? In the nucleus. Then a copy is made. Yep, we call that mRNA, messenger RNA. We'll learn that next chapter. That copy is sent to the rough, the rough ER. And it's the ribosomes here. The ribosomes that are attached, that's what's going to scan that copy, that mRNA. And it's going to translate the DNA language to an amino acid language.
I'm sorry.
Simply
So the answer to 27 is messenger RNA.
No, the answer right here is rough ER.
To number 27, rough
27
ER.
is rough, right here in this blank right here, rough. And then the second blank is ribosome, or ribosomes, plural.
And what was 26? Sorry.
Yeah, 26 was nucleus right here.
Okay.
Yes. So they're trying to show you the trend. Nucleus to the rough ER. That's the trend. And where were ribosomes originally made? Right here on this third blank. Nucleolus, perfect. So now that the rough ER has made the protein, where is it going to go?
The Golgi apparatus.
Protein, that newly formed protein pump to the Golgi apparatus or Golgi complex, which is going to package it into a nice little vesicle, which is its own little phospholipid bilayer. It's so cute. And then it's going to push that vesicle, it's going to push it right against that cell membrane. And either that chloride pump becomes part of the membrane in this case, which is what's going on, or it could be exocytosis if it were a hormone or enzyme. But in this case, it's going to be a chloride pump. So it embeds it right there in the membrane. And that's how cells maintain themselves. They build proteins every single day, all day long. That's what keeps your cells doing what they do. Constantly building, constantly rearranging themselves, rehabilitating themselves best they can, given the environment that we give them. That's why you got to pick days of the week where you're really good to your body. Fruits and veggies, all fresh fruit and veggies, right? No packaged foods, no soda. It's tough, but do your body a favor, at least a couple of days a week. Please don't drink soda every day. I know it's a habit, but You'll thank me when you're older. Okay, lastly, these last few organelles. So number 29, where's 29? There we go. That's going to be a lysosome right there and right there. And the other one here is peroxisome in the white. So number 30 is a mitochondria, 31 is the centriole. And just get those functions from your PowerPoint slide to make that easy. For this last case study, it's simply just lysosomes. Whenever there's a buildup of a waste product like cerebroside, that's a waste product. It's supposed to be broken down. Well, guess what breaks down things in the cell? Lysosomes. So this is a problem where the lysosomes either are not producing the digestive enzymes or they're not being made properly. Either way, that's what's going on in Gaucher's disease. The organelle, the lysosome, is dysfunctional. So it does not properly break down or detoxify the cell. Both lysosomes and peroxisomes detoxify, but lysosomes have a bigger job of breaking down all kinds of waste products like this one. So let me just shoot to the PowerPoint one more time because we are done, guys. Just to clue you in on where those case studies are, they're at the very end, last 3 slides. Whoops, let me do it this way. There we go. So if the picture bothers you, don't pay attention to it. Just look at the clinical impression. That's what you need to know from these case studies. That'll help you on the test. For jasmine, with more aquaporins, increased water reabsorption. Kind of goes hand in hand, allowing the body to retain more water, both inside the cells and outside the cells, because we all know that pregnant women get swollen, which is why you got to keep track of the blood pressure. That's very important. Don't want that to get out of hand. And for cystic fibrosis, check out down here. Without effective chloride pumps, the extracellular compartment becomes dehydrated and the mucus thickens. Our last one, Gaucher's, without effective lysosomes, metabolites, which are waste products from the energy in the cell, are not broken down due to a buildup of the waste products, or excuse me, causing a buildup of waste products, which toxifies the cell. Not a good environment. So there you go, guys. That is everything we've got for this lab. There is no quiz for this, but there are the 2 Connect assignments, the homework and the quiz for Connect. But that would do it for this lab, and you'd be ready for your tests, hopefully. So as far as new material goes for this lab, kind of straightforward. The new material is just really identifying the organelles and listing their function. And then you got these 3 case studies. And then you're really just confirming that you know the structure of the cell membrane, understanding which is the nonpolar and polar regions, philic and phobic. So reviewing this video or reviewing that worksheet that we just finished would be a great study guide for this lab. Any last questions? Otherwise, we are done. You can go do your other homework if you'd like.
Thank you.
Okay, thanks for staying a little later today. I know it was a big day. Best of luck on everything with the exam, but I'll see you in lecture tomorrow if I've got you.
Where are the case studies? I can't find this that information in my PowerPoints that I have printed out.
It is the last 3 slides of Lab 4.
Okay. Okay. I guess I have not downloaded and printed out Lab 4 because
no, you've got it. The first PowerPoint that I uploaded was actually Labs 1 through 5, but since this semester we changed things up, I went ahead and edited it just to be clear. So Labs 1 to 4 is now on the updated PowerPoint. So go check that out.
I will do that. Thank
Yep.
you.
Okay.